Hello friends, welcome back to a new video, that is MCQs on biotechnology, and this is the part 4 of this series of videos on biotechnology. So the first question of this video, that is genetically modified organisms are also abbreviated as option A GMO, option B MOG, option C GOM, or option D all of the above, and the right one is option A, that is GMO. So genetically modified organisms, that is GMO, are abbreviated as GMO. Okay, so GMO is abbreviated as genetically modified organisms. Okay, so let's move on to the next question, that is question number 62. That is, which of the following is or are methods of gene amplification techniques? And the options are PCR or ligase chain reaction, that is LCR, or SSR, that is self-sustained sequence replication, or option D, that is all of the above. So the right answer is option A, that is PCR. So PCR is the method which is used for gene amplification. Okay, so let's move on to the next question, that is question number 63. So the biotechnologically derived rice from Oryza sativa with large beta-carotene content called golden rice was first created by option A Potricus, option B Peter Beer, option C both A and B, and option D Sanger and Nicholson. And the right answer is option C, that is both A and B. That means Potricus and Peter Beer are the right answer. So the biotechnologically derived rice from Oryza sativa with large beta-carotene content called golden rice was first created by option A and B, that is Potricus and Peter Beer. So let's move on to the question number 64. That is, transgenic plants can act as living bioreactors for the production of chemicals and pharmaceuticals. This is known as agriculture farming, molecular farming, pharmacotechnology, or organic farming, and the right one is option B, that is molecular farming. So the transgenic plants can act as living bioreactors for the production of chemicals and pharmaceuticals. So this is known as molecular farming. Okay, so let's move on to the question number 65. So which of the following organisms were applied for creating golden rice? And the options are Agrobacterium tumefaciens, option B Arbuenia iridovora, or option C both A and B, or option D neither A nor B. So the right answer is option C, that is both A and B are the right answer. That is the A and B, that is Agrobacterium tumefaciens and Arbuenia iridovora are the organisms which are applied for creating golden rice. Okay, so let's move on to the question number 66. That is, regulatory sequence elements can be identified by option A reporter gene assay, option B primer extension, option C both A and B, option D none, and the right one is option C, that is both A and B. So regulatory sequence elements can be identified by reporter gene assay and primer extension, that is both A and B is the right answer. So let's move on to question number 67. That is, overlapping DNA sequences can be identified by option A SAGE, that is SAGE, or option B clone fingerprinting, or option C chip assay, or option D 2D page. So the right answer is option B, that is clone fingerprinting. So overlapping DNA sequences can be identified by clone fingerprinting. Okay, so next question देखेंगे. Next question is question number 68. So a target gene can be replaced by option A homologous recombination, option B clone fingerprinting, option C zoo blotting, option D fish method. So the right answer is option A, that is homologous recombination. So the target gene can be replaced by homologous recombination. So next question अभी क्या है? Next question is, the protein having complete amino acid sequence formed from two or more proteins from different gene product is referred as option A green fluorescent protein, option B fusion proteins, option C RAPD protein, option D xenoproteins, and the right answer is option B, that is fusion proteins. So the proteins having complete amino acid sequence formed from two or more proteins from different gene products is referred as fusion proteins. Okay, so question number 70 देखेंगे. Question number 70 is the copying of segment of genes within the DNA or RNA molecules is called option A gene stacking, option B gene banking, option C gene amplification, option D gene filtration. So the right one is option C, that is gene amplification. So the copying of segment of genes within the DNA or RNA molecules is called gene amplification. So question number 71 देखेंगे, that is the scientific study of genes and their role in an organism structure, growth, health, disease, etc. is referred as option A gene analysis, option B genomics, option C proteomics, option D gene banking, and the right one is option B, that is genomics. So the scientific study of genes and their role in organism structure, growth, health, disease, etc. is referred as genomics. So let's move on to the question number 72. So the insertion of antisense DNA molecule in vivo into selected cells of the body in order to block the activity of undesirable gene is called option A gene therapy, option B gene targeting, option C gene mutation, option D transition gene targeting. So the right answer is option B, that is gene targeting. So the insertion of antisense DNA molecule in vivo into selected cells of the body in order to block the activity of undesirable gene is called gene targeting. So let's move on to the question number 73. So the selective deliberate manipulation of genes or genetic material by man is called as option A gene therapy, option B gene library, option C genetic engineering, or option D it is impossible. So the right one is option C, that is genetic engineering. So the selective deliberate manipulation of genes or genetic material by man is called as genetic engineering. Okay, so let's move on to question number 74. So which of the following is or are applications of gene expression analysis system? And the options are differentiate between normal cell versus cancerous cell, option B differentiate between normal cells versus cell receiving stimulus, or option C, that is differentiate between cells from same organisms at different stages of development, or option D all of the above. And the right one is option D, that is all of the above are right. So all of the above conditions are applications of gene expression analysis system. Okay, so let's move on to question number 75. That is the phenomenon of suppression of gene expression is referred as option A gene cloning, option B back mutation, option C gene silencing, option D gene divergence, and the right one is option C, that is gene silencing. So the phenomenon of suppression of gene expression is referred as gene silencing. So let's move on to question number 76. That is what happened when wheat field is inoculated with rhizobium? Option A no increase in production or nitrogen content of soil, or option B fertility of soil increase, option C fertility of soil decreases, or option D increase in production and nitrogen content of soil. So the right answer is option A, that is no increase in production or nitrogen content of soil. So what happened when wheat field is inoculated with rhizobium? No increase in production of no increase in production or nitrogen content of soil. So next देखेंगे, next is 76, that is which of the following is or are methods of gene amplification techniques, and the options are PCR, Q beta replicase techniques, option C strand displacement amplification, that is SDA, or option D all of the above, and the right one is option D, that is all of the above are right. So which technique of the following is or are methods of gene amplification technique? So all of these, that is PCR, Q beta replicase technique and strand displacement amplification, all are the methods of gene amplification techniques. Okay, so let's move on to next question, that is question number 78. So post-zygotic sexual incompatibility can be overcome by option A protoplast fusion, option B ovule culture, option C in vitro pollination, option D bud pollination. So the right answer is option A, that is protoplast fusion. So post-zygotic sexual incompatibility can be overcome by protoplast fusion. Okay, so the next question is which one of the following does not apply to sporophytic self-incompatibility system, and options are 3-celled pollen, option B dry stigma, option C rejection reaction in the stele, option D in vitro pollen germination is difficult, and the right one is option A, that is 3-celled pollen. So which one of the following does not apply to sporophytic self-incompatibility system, that is 3-celled pollen. Okay, so the last question of this video, that is the set of general methodologies, practices, protocols and regulations mandated from Food and Drug Administration for testing and manufacturing pharmaceuticals is referred as option A GMP, option B WHO, option C GLP, option D none of the above, and the right answer is option A, that is GMP. Okay, so GMP is the right answer for the set of general methodologies, practices, protocols and regulations mandated from Food and Drug Administration for testing and manufacturing pharmaceuticals is referred as GMP, that is good manufacturing practice. Okay, so these are all 20 questions of this video, that is the part 4 of this series of videos on biotechnology. Okay, so thank you for watching this video.